So let's jump to an example and talk about how early life experience epigenetically programs the stress system. To briefly review, when our body encounters a stressor, it activates the hypothalamic pituitary adrenal axis, which results in the release of cortisol in the body, which has a variety of adaptive effects in the short term and is not terribly adaptive in the long term. So to keep this response short and sweet, there are negative feedback systems in the brain that serve to truncate the response, which terminates the cortisol release from the adrenal gland and resets the system for the next stressor. In certain clinical populations, this system can actually go awry, and so the example that we will talk about is what we see in patient populations that have been exposed to early life trauma or stress. In these adult patients, we see that the same type of stressor, while it activates the HPA axis and results in cortisol release, there's a lack in the negative feedback mechanisms. And so what happens is you get this prolonged and more intense cortisol response, and the system has difficulty resetting. And it's this prolonged hyperarousal state that's thought to relate to the increased risk we see for borderline personality disorder, PTSD, and some affective disorders in patients that have experienced early life trauma or neglect. And this is really a fundamental question in psychiatric neuroscience. How does an early life experience like trauma or neglect biologically program a circuit in a clinically relevant way that lasts throughout the lifespan of the person? And yet, despite being a critically important question, it's actually really hard to answer in humans. We're so psychosocially complex that isolating the biological mechanisms is not an easy task. And so Michael Meaney's group decided to jump to a different species that's arguably a little simpler from the psychosocial perspective, and that was by looking at rat maternal behavior. And so his group started off with a very simple question, which was, is all rat maternal care created equally? And it turns out it's not. There's actually a normal distribution of maternal care in rodents. And you might ask, how does a rodent show that it loves its little rodent children? And it turns out that licking and grooming is how rat moms show that they really love their rat pups. And so what his group found is that while most rats are sort of good enough mothers, there are two extremes in the licking and grooming categories. One, which are these amazing, wonderful rat mothers who just love their pups like gangbusters. And then there's sort of this other extreme of the spectrum, which are these sort of sad, not so caring mothers that really only sporadically lick and groom their pups. So a high licking and grooming experience with lots of love on one extreme, and a low licking and grooming experience on the other extreme. So what do these babies actually look like? Well, much like the human condition, in the loving, high licking and grooming environment, we see low stress reactivity, so a system that's able to react to stress but then reset. And in this low licking and grooming group, we actually see this increased stress reactivity, prolonged corticosterone responses, and just overall difficulty truncating that response. So very much like the human population that we talked about earlier. So now the question becomes, is it actually the experience that's programming the stress system in these animals? And so there's two experiments that were done to really get at that question. And the first is sort of these classic cross-fostering experiments, which are like little rat adoption agencies. So when you take the newborn pup from the low licking and grooming mom and have her adopted by the high licking and grooming mom on the first day of life, we find that she ends up looking a lot like the biological children of the high licking and grooming mom, and vice versa. So taking a high licking and grooming pup on the first day of life and giving her to this low licking and grooming, not so caring mom, we see the phenotype of the low licking and grooming offspring. So that's one piece of evidence, but Meaney's group actually took it a step further and wanted to show that it's actually the tactile experience and not something else about the environment of being with the high LG moms. And so they did an experiment using paintbrushes to actually mimic the experience of being licked and groomed. So you can imagine some poor graduate student sitting in the vivarium for the entire first 7 days of life with a paintbrush trying to do this experiment. But the results are really exciting, and that's that simulating the high licking and grooming environment, these are my paintbrushes, in a low LG pup for the first 7 days of life recapitulates the high LG experience. And so you see as adults, those rats have lower stress reactivity, and vice versa, taking a high licking and grooming pup and only giving them a little paintbrush love recapitulates the low LG phenotype with this heightened stress reactivity. So what these experiments tell us, what this poor graduate student in the vivarium with a paintbrush is leading us to, is the point that experience matters. And this is probably not a shock to the clinical audience. But now, using this model system, we actually get to the really cool part, which is, how does this work? And to answer that question, we go to the neuroanatomy. So what do the brains of these animals look like? So here again, we have the low LG animals on the left and the high LG animals on the right. And you can see my incredibly sophisticated artist rendering of hippocampi in both of these animals. So Meany's group decided to look at glucocorticoid receptor expression in the hippocampus. So glucocorticoid receptors are those that actually sense these stress hormones and thus are well poised to regulate neural responses to them. And what they found was that in the high LG animals, you saw a high density of glucocorticoid receptors in the hippocampus. In contrast, in the low LG animals, they saw a much sparser distribution of glucocorticoid receptors in the hippocampus. And why do we care about this? We care about this because the hippocampus projects to the master regulator, the H and the HPA axis, and actually provides that negative feedback. And it's been shown that this high density of glucocorticoid receptors is associated with a very robust negative feedback response, whereas the low density of glucocorticoid receptors is associated with sort of a wimpy negative feedback response. And so these feedback responses make a lot of sense when we think about the phenotypes of the animal. So this high stress reactivity in the low LG animals because of a wimpy negative feedback response versus low stress reactivity in the high LG animals with this very robust, tightly regulated negative feedback response. And so now we get to the final piece of the puzzle, which is how do we get from this early life experience to glucocorticoid receptor expression? And this is where the epigenetic part comes in. So Meany's group decided to take a look at the glucocorticoid receptor gene and specifically the epigenetic decorations on the promoter region. And what they found was that at birth, everybody's promoter is heavily methylated. And if you remember from the first segment, heavy methylation of the DNA is associated with low transcriptional activity. So what impact does early life experience have on these epigenetic markers? Well, in the low licking and grooming animals, this promoter remains methylated for the life of the animal. And so this heavy methylation is associated with low transcriptional activity, low glucocorticoid receptor expression, and low negative feedback to the HPA axis. In contrast, high licking and grooming actually causes a demethylation event at this promoter, removing the obstacle to the transcriptional machinery. Transcription factors like NGFIA are now able to bind. You get robust transcription of the glucocorticoid receptor, high expression in the hippocampus, and tightly regulated HPA responses. So lastly, I'll just tie this back to the clinical population by mentioning that there have been studies done now in humans with postmortem brains looking at epigenetic signatures of the GR gene and expression in the hippocampus, and they essentially recapitulate the findings that we've described for the rat. So in human beings that have been exposed to trauma or early life neglect, we find heavily methylated promoters, low GR expression, which correlates with the phenotype of poor negative feedback and high stress reactivity, whereas controls that have had no trauma show demethylated promoters, high gene expression, and efficient regulation of the stress system. So the take-home points are, one, that experience matters, that experience shapes our brains, but two, that the study of epigenetics helps us understand how that process occurs on a biological level, and better understanding this system allows us to develop novel therapeutic targets and therapies that will help our patients.